What if we could spot cancer way earlier than ever before? And what if we could do it not with some invasive, complex biopsy, but from a single drop of blood? Well, today, that's exactly what we're talking about. A totally revolutionary approach that combines these tiny biological messengers with the massive power of AI and some brilliant miniature engineering.
Okay, let's just start with the one fact that really drives all of this. It's something we all know, but it's so important. When it comes to cancer, the sooner you find it, the better the odds. It's that simple. And that powerful truth is the whole reason behind the incredible science we're about to get into.
So our guide through this is a really groundbreaking review put together by a team of researchers, Sanjita Muthamilselvan and Ashok Palaniappan from SASTRA University in India, working with Priyanth Ramasamy Sundar Baabu over at Sungkyunkwan University in South Korea. What their work does so brilliantly is connect three totally different fields of science into one single powerful idea.
So here's how we're going to break it down. First, we'll look at these tiny messengers that are basically leaving clues all over our bodies. Then, we'll see how AI can sort through all those clues to find the real patterns. After that, we'll explore the tiny lab-on-a-chip that makes the actual testing possible. And finally, we'll take a step back and see what this all means for the future. You ready? Let's get started. All right, so our story starts at the microscopic level. I mean, we're talking about some of the smallest but also most powerful molecules that exist in our bodies. These are the secret clues that tumors are constantly leaving behind for us to find. So what are these things we're actually looking for? Well, they're called microRNAs, or miRNAs for short. And the best way to think of them is like tiny little master switches. See, they don't actually build anything in your cells, but they're the ones in charge of telling your genes when to turn on and when to turn off. They're absolutely essential for keeping everything running smoothly. Which, of course, leads to the big question, what on earth do these tiny little switches have to do with a disease as massive and complicated as cancer? Well, it turns out everything. Because in cancer, these switches start going haywire. You see, tumors are constantly shedding their own broken, dysregulated mRNAs right into our bloodstream. And that's where they become clues. Things like tumor invasion or building new blood vessels to feed itself. Those processes are driven by specific mRNA signals. And because those signals leak out, we can perform a liquid biopsy. Instead of actually having to cut out a piece of tissue, we can just take a simple blood sample and basically listen in on what the tumor is up to. And these signals aren't all the same. They come in different flavors, you could say. You've got your oncomers, which are like stepping on the gas pedal for tumor growth. Then you have the tumor-suppressing mRNAs, which get silenced. That's like cutting the brakes. There are even specific ones for helping cancer spread, or build its own blood supply, or even hide from your immune system. Each one tells a crucial part of the story. Okay, but here's the huge challenge. Finding these things is like looking for a needle in a haystack, a really, really big haystack. These circulating miRNAs make up only about 0.01% of all the RNA floating around in your body. It's an incredibly faint signal. So, how in the world do you find it in all that noise? Well, you bring in a detective, one with like superhuman pattern recognition abilities. You bring in artificial intelligence. I mean, think about it. In just one blood sample, you could have thousands of different mRNA signals flying around. Some are totally normal, some are just random noise. And buried in there somewhere are the critical biomarkers for a specific cancer. There's no way a person could sort through all that. But for an AI, that's just a data problem waiting to be solved. And the process for teaching the AI is pretty methodical. First, you feed it just enormous amounts of data, mRNA profiles from thousands of samples, some from healthy people, some from people with cancer. Then the AI gets to work. It sifts through everything to find what they call the most important features. That's the small handful of mRNAs that are the most predictive. From there, it trains itself to recognize that specific cancer signature, and finally, it's tested on brand new data it's never seen to make sure it's accurate. And the AI can do two incredibly important jobs here. The first is diagnosis, right? Just answering the question, is cancer there? And if it is, what kind is it and what stage is it at? But the second job is just as critical, prognosis. The AI can look at that mRNA signature and help predict how aggressive the cancer is likely to be. That lets doctors sort patients into high-risk or low-risk groups, which is a huge step toward more personalized treatment. And building one of those prognostic models follows a pretty similar logic. The AI looks at patient data and hunts for the specific mRNAs that are linked to survival outcomes. Then, it narrows that list way down to just the absolute most critical ones and builds a formula,
a risk score. And that score becomes a powerful tool that can help guide really important treatment decisions. It's a game changer.
Okay, so we've got the biological clues, the mRNAs, and we have our super detective AI that can identify the specific signature we need to look for. But how do you actually turn all of that digital insight into a physical test that a doctor can use in a clinic?
Yeah, this is the final and maybe most important piece of the whole puzzle. It's one thing to have a powerful AI model running on a computer somewhere, but it's something else entirely to have a fast, cheap, and easy-to-use test. The technology that builds that bridge is called microfluidics.
So what is microfluidics? Put simply, it's the science of building tiny, tiny plumbing systems. We are talking about channels that are smaller than a human hair. And by controlling these tiny little drops of fluid with incredible precision, you can basically shrink an entire complex laboratory process down onto a single small chip. It's quite literally a lab on a chip.
And this is where it all just clicks together perfectly. You see at the top, the AI does all the heavy lifting up front. It uses machine learning to sift through mountains of data and discover the perfect biomarker signature. Then, that exact signature gets engineered into the microfluidic device you see at the bottom. So now when you add a new patient's blood, the chip isn't searching blindly anymore. It's been programmed to look for one very specific thing and one thing only.
And the performance of these little chips is just mind-blowing. I mean, look at these numbers. We are talking about detection limits in the attomolar and femtomolar range. That is such a ridiculously small concentration. It's like trying to find one single specific grain of sand from all the beaches on planet Earth. And these chips can be designed for all sorts of cancers using all kinds of different tech, from nanoparticles to CRISPR.
So you can see why this is a complete game changer. You only need a tiny drop of blood, you get incredibly sensitive results, and you get them fast. Plus, the devices themselves are low cost and portable. This is how you move high-tech diagnostics out of a big, specialized lab and get it closer to where patients are actually being treated.
So when you start to put all of these pieces together, the biology, the AI, the engineering, what does the future of medicine even start to look like?
I think this quote, pulled directly from the researcher's paper, says it best. Their ultimate goal is to pave the way for accessible, precision, personalized medicine. And those three words are so key. Accessible, meaning for everyone. Precision, meaning for the specific disease. And personalized, meaning for the individual patient.
What we've really seen today is just this beautiful convergence of science. You need the deep expertise of biology to even understand what mRNAs are. You need the raw power of data science and AI to find the patterns, and you need the sheer ingenuity of engineering to build the actual device. And when you bring them together, you create something that no single field could have ever achieved on its own.
And that leaves us with a final pretty exciting thought. We focused on cancer today, but this core idea of finding a biomarker signature and then loading it onto a chip, that could work for countless other diseases. So I'll leave you with this question. What could we achieve if a simple, accurate test for dozens of diseases could fit in the palm of your hand?